So friends, we were discussing about the factors that affect somatic embryogenesis, and in that we discuss about the effect of growth regulators on somatic embryogenesis. Some of the growth regulators like auxins, they prevent or they inhibit the conversion of pro-embryogenic masses into embryos, whereas certain growth regulators like cytokinins, brasinosteroids, they promote somatic embryogenesis. So the next factor is the selective subculturing. Now we know that explants are multicellular in many cases, and they contain mixture of cells which differ in their potential to form somatic embryos, because every cell or which is under in vitro condition will behave differently depending upon its capacity or ability to develop. So the culture of such explants is also heterogeneous. Obviously, if we are using a multicellular explant, we are culturing that multicellular explant under in vitro conditions. The culture which we will develop will obviously will have the heterogeneous nature in terms of the capacity of the cells to develop into embryos, and therefore selective subculturing is always recommended to increase the chances of successful somatic embryogenesis. Now, from the callus or the suspension culture which has been developed by using multicellular explant or any other suitable explant material, the cells which are having higher propensity or the potential to get converted into somatic embryos should be separated, and then such cells should be further cultured to develop into somatic embryo to withstand the somatic embryogenesis process. Such kind of separation of those cells having higher propensity to get converted into embryos, we call it as selective subculturing. So if we are doing selective subculturing during the somatic embryogenesis process, then it will enhances the chances of a successful process, successful conversion of somatic cells into embryos. Now, embryogenic callus cells can be identified morphologically. We can easily identify such kind of cells which are having greater propensity to get converted into embryos. I have taken certain examples like the species of Poaceae family, the embryogenic callus appeared granular with a smooth surface, whereas the non-embryogenic cells in the callus were rough, friable and translucent. Similarly, the embryogenic calli of Coffea arabica were found to be brown and hard, while the non-embryogenic calli of the coffee were pale in color and were very friable. So these are only couple of examples which show that how you can identify the embryogenic callus cells during the subculturing, and you can use such embryogenic cells to convert them into somatic embryos. The next factor is the use of electrical stimulation. It has been found that mild electric current always stimulate the shoot and embryo differentiation. It has also been found that exposure of the protoplast to low electric field enhances direct embryogenesis from 40 to 100%. So you can just imagine there is an increase in the success rate from 40 to 100% when the protoplast were exposed to low electric field, and the number of embryos per plate was increased from 76 to 116. But the electrical stimulation has been found to cause changes in the organization of microtubules, which eventually affects the cell polarity, thereby causing the beneficial effects towards somatic embryogenesis. But one has to be very critical while using electric stimuli. One has to optimize the strength of the electric current which is used to expose the cells to initiate the embryogenesis. Next factor is the induction of stress or exposure of stress towards the cells. Now, it has been found that saline conditions and high sucrose concentration always promotes the somatic embryogenesis. Presence of heavy metals obviously under appropriate concentration below a particular threshold value always cause a stress that will promote somatic embryogenesis. But yes, the concentration of heavy metals beyond a threshold value will have a negative impact on the somatic embryogenesis. It will lead to the toxicity of the cells. Then temperature. Now, the exposure of the temperature for a suitable period of time will depend or will vary from species to species. Some plant species cells, they prefer exposure to higher temperature, whereas certain cells of certain species will favor lower temperature exposures for shorter period of time or longer period of time. So, one has to again optimize the temperature condition in order to induce the somatic embryogenesis or in order to promote somatic embryogenesis. Nutrient starvation, wherein we expose the cells towards deficiency of nutrition. Again, such kind of stress helps in promoting somatic embryogenetic, promoting the or the upregulation of the genes that are responsible for conversion of cell into somatic embryos. So, nutrient starvation stress always has been found beneficial for somatic embryogenesis. Now, there are certain factors that also affect other factors that affect somatic embryogenesis, like cellular density in the media. It has been found that high cellular density will increase the chances of release of high amount of phenolic compounds as the byproducts of the metabolic processes. And these phenolic compounds have been found to enhance the stress on the cells to such an extent that it inhibits the somatic embryogenesis. Then high concentration of oxygen in the media decreases somatic embryogenesis because it fasten up the germination process, whereas if we decrease the oxygen concentration in the media, it promote somatic embryogenesis, because decreasing the oxygen concentration will slow down the germination rate, vis-a-vis it also enhances a sort of stress onto the cells, which again promotes the somatic embryogenesis. Another factor is light. The intensity and the type of light, the effect of intensity and type of light on somatic embryogenesis will vary from cell to cell or the plant species to plant species. By and large, it has been observed that green and red light increases the somatic embryogenesis, while white light decreases somatic embryogenesis. Now we move on to the last aspect of somatic embryogenesis, that is applications. Now we develop somatic embryos from the somatic cells, and therefore such kind of somatic embryos help in clonal propagation and mass multiplication. So since we are using the pre-embryogenic cell or any other suitable explant material to get converted into somatic embryos, such kind of somatic embryos will retain the genetic or genome uniformity, and therefore they are helpful in clonal propagation. Somatic embryogenesis are widely used for the production of synthetic seeds, which I am going to discuss in detail in my subsequent lecture. Then genetic transformations can be done by using somatic embryogenesis with the help of agrobacterium or any other suitable media or the process by which we can introduce the desirable genes into those somatic embryos. Somatic embryos are also used in germ plasm conservation of certain high value plants like sandalwood is there, coffee is there, or these are also used in conservation of certain endangered plant species. Somatic embryos are also used for pathogen elimination, that is, we can produce virus free plants by using specific somatic embryos. It is also used in commercial forestry to increase the population of a particular, specifically in case of pines. So, to increase the population in a specific area, somatic embryos are widely used. And last but not least is the mutation breeding and soma clonal variations. So, if you want to introduce any kind of changes in the genetic makeup of a plant, we can introduce that variation in the somatic embryos. You want to introduce a desirable variation in the genome, you can always go with that kind of variation by using somatic embryos. So friends, these were the applications of somatic embryos or somatic embryogenesis. Now we move on to the synthetic seeds. As we discussed that somatic embryos are largely used in the production of synthetic seeds. So what we are going to learn about synthetic seeds is the brief historical development, then what are synthetic seeds and what are different types of synthetic seeds. How we can prepare synthetic seeds either by using a suitable explant material or by using the somatic embryos which we have developed. And last would be the applications of the synthetic seeds. So coming on to its historical development. It was Stewart along with his team in 1958 and Rennert in 1959 who initiated the somatic embryogenetic process, which laid the foundation of the formation of synthetic seeds. So what they did was they basically prepare the somatic embryos from the carrot tissues and they encapsulated those somatic embryos. But this process was, as I told you in my previous lecture, this whole process was very laborious and costly, and therefore majority of the scientific community lose interest in the development of synthetic seeds until 1977 when Murashig encapsulated the somatic embryos into alginate beads or the alginates, and he defined synthetic seeds as a one line, he gave a one line definition that encapsulated single somatic embryos, and he called them as synthetic seeds. Now after this successful experiment, the interest in synthetic seeds again regenerated and lot of scientists start working in the field of synthetic seeds. Drew in 1980 again he developed commercial propagation using somatic embryos. He developed synthetic seeds using somatic embryos from the carrot. But again the commercial propagation was not very much successful. Later on, Gray et al. In 1981 they developed a successful commercial propagation technique by using synthetic seeds. Keto and Genic in 1982, they for the first time developed hydrated synthetic seeds again using carrot embryos by using polyethylene glycol. Keto and Genic again in 1985 use another polymer, that is polyoxyethylene, for encapsulation of the somatic embryos. So this is how the development or the encapsulation process of the somatic embryos or the development of synthetic seeds from somatic embryos, the process was developed. Various polymers, new polymers were developed, and significant progress was made in the overall process. Now we move on to the actual definition of synthetic seeds. Now as the name seed indicates, these are nothing but these are the sources by which a new plant can be developed. When we use the term synthetic, it means we develop such kind of seeds under in vitro conditions. So the simple definition of synthetic seeds could be artificially encapsulated somatic embryos or any other suitable explant material like shoot bud, cell aggregate, or any other tissue that may be utilized for planting as a seed and that has the capacity to germinate under in vitro circumstances and that retains its capacity even after preservation. So if we talk about simple seeds, they are nothing but they are the agents that help in development of a new plant. But synthetic seeds, we encapsulate the suitable explant material, which can be somatic embryo, which will give you a bipolar development both root and shoot, or any other suitable explant material like shoot buds, which we have discussed, which will give you a unipolar development until and unless you expose them to a specific cultural condition. In addition to their ability to develop into new plant, they should retain that capacity to germinate under in vitro condition and they should retain the capacity even after preservation. So this is the crux of the definition of synthetic seed: suitably encapsulation of suitable explant material which retain the capacity to germinate under in vitro condition and retain the capacity even after preservation. Now if we compare the synthetic seeds and the natural seeds, we know that natural seeds are formed from the germ cells, whereas synthetic seeds can be formed from any vegetative cell. We know that natural seeds are produced by sexual process, whereas synthetic seeds can be produced by the asexual process, and the formation of natural seeds involves the fusion of gametes, whereas in case of synthetic seeds no fusion of gametes is involved. Since fusion of gametes is involved in natural seeds, it contains genes from both the parents and there is a recombination of the genes. Whereas in case of synthetic seeds, no fusion of gametes occur, therefore genes of single parent is present in the synthetic seed and there is no genetic recombination in these kinds of synthetic seeds. Now, if we look into the structure of synthetic seed, we can break the structure of synthetic seed in two major parts. First is the explant material, the material which we are going to encapsulate and will the material which will develop into a new plant. And the second part is the capsule in which the explant material is encapsulated, and this capsule will be having gelling agent, different types of nutrients to allow the explant material to retain the capacity to germinate, to retain the capacity to develop into a new plant. Various antibiotics or antimicrobials could be there which will help in preventing any kind of microbial infection. So, coming on first to the explant material. Now, explant material can be bipolar like somatic embryos or embryogenic cells, or explant material can be unipolar, which we will discuss further, like shoot buds are there, root buds are there. So, coming on to the bipolar explant material, and the most widely used bipolar material is the somatic embryos. Now, they have a specific advantage why somatic embryos are always preferred in manufacturing of synthetic seed, because the plant lines keep their high regenerative capacity for long time. Further, these somatic embryos also exhibit a great deal of genetic stability and less somaclonal variations are there. So, genetic uniformity can be maintained if we use somatic embryos in preparation of synthetic seeds. But the major disadvantage of using somatic embryo is the late onset of root formation has been observed if somatic embryo is used as an explant material. Then another type of bipolar explant material that can be used is the embryogenic tissues. And embryogenic tissue can be an ovule, can be any male gamete which has a propensity of developing into embryo cells. Now, this can be these embryogenic tissues are largely used when you want to proceed with clonal propagation and you want to study the genetic transformations into the plant cells. The major disadvantage of using such kind of embryogenic tissue is the maintenance of these embryogenic tissues is quite laborious and is very costly in comparison to the somatic embryos. If we talk about the unipolar explant material, as we discussed, you can use shoot or root tips, nodal segments, apical or axillary buds, you can use micro shoots, you can use protocombs can be used, which are mainly used for orchids. The major advantage of unipolar explant material is the uniform production of the plants and can be used, and such kind of unipolar explant materials can be used for both hydrated as well as desiccated synthetic seeds, which we are going to discuss in detail later on. Then the major disadvantages: asynchronous growth and late maturation, because these shoot tips or root tips would be multicellular explant material and therefore there will be an asynchronous growth. So, these were certain advantages and disadvantages of the unipolar explant material. Now, we move on to the second component of synthetic seed, that is capsule or the matrix, and we discussed that it largely it is made up of the encapsulating agent, the agent which is used to encapsulate the explant material. The most widely used gelling agent is the alginates because of their appropriate thickness and weak spinnability. Now, spinnability is a property by which the gelling agent, when it is exposed to a pressure and is allowed to pass through a narrow orifice, it will form a filamentous structure. Now, this formation of filamental structure is not desirable in case of synthetic seeds. Therefore, the agent which are used to encapsulate the explant material should always have low spinability, which will allow this gelling agent to form suitable size beads. Such kind of gelling agent or alginates which are most widely used, they have low toxicity, they are cost effective, they are compatible with the most of the explant material, and they have fast gelation rate. And that is why alginates are the most widely used encapsulating agents or the gelling agents for the production of synthetic seeds. Apart from alginates, agar, polyoxyethylene, carrageenan, guar gum, sodium pectrate, tragaganth, gelem gum, etc. There are so many other agents are also used. Now, apart from gelling agents, complexing agents are also used like calcium chloride, potassium chloride, ammonium chloride, and these complexing agents are largely used for hardening of the gelling beads or the gel beads that are formed by encapsulating the explant material into the gelling agents. We will discuss the mechanism of the complexing agents later on. Then third component is the matrix or the simulated endosperm, which will provide all the nutrients to the explant material to retain its ability to grow, to retain its ability to get converted into the plant. And this matrix is composed of suitable amount of nutrients, growth regulators, antimicrobials, pesticides and fungicides, and activated charcoal. Now, activated charcoal helps in absorption of certain toxic released by the cells, as well as it also absorbs certain nutrients and will allow a sustained release of the nutrients in the media. Now, we move on to the types of the synthetic seeds, and we discussed broadly there are two types: desiccated synthetic seeds and hydrated. Now, desiccated synthetic seeds are used for the desiccant tolerant plant embryos, those embryos which can tolerate desiccant conditions. Now, these were reported for the first time by Keto and Genick, and they encapsulated the embryo in polyoxyethylene glycol to encapsulate the explant material, and then they subject those encapsulated material towards desiccation, either by placing them overnight under laminar flow or by using any other suitable condition under controlled environment. Now, desiccation tolerance can also be induced by exposing the embryos to abscisic acid or suitable osmoticants, or by sublethal doses of different types of stresses like nutrition deprivation or low temperatures. But all these factors, all these exposing or the strategies to induce or to make embryos desiccation tolerance, one has to optimize each and every strategy that is to be used to desiccate that embryo. Second type is the hydrated synthetic seed. Now, this is largely used for the desiccant sensitive plant embryos, those plant embryos which cannot withstand the desiccant conditions, and therefore we encapsulate such kind of explant material or plant embryos into suitable media or hydrogels like alginates or agar, which we have already discussed. Now, such kind of encapsulation provides protection to these somatic embryos towards different kind of mechanical or shear or physiological stresses. Now, if we look into the preparation of synthetic seeds, there are three major steps. First one is the selection of suitable explant material, whether you are selecting a somatic embryo or you are using a unipolar tissue. One has to optimize or use suitable explant material depending upon the goal of the production of synthetic seeds. Second step is the encapsulation of the explant material, and third is the complexation of the encapsulated explant material by dropping it into the complexation solution. Now, the last two steps largely encompass the encapsulation process. Now, if we further break down encapsulation, it has three steps: coating, complexation and rinsing. Now, coating is nothing but embedding the explant material into the gelling agent, which is largely sodium alginate is used in the range of 2 to 5%, and other alginates, other gelling agents we have already discussed. Second is the complexation. Now, once we have placed the or embedded the explant material into the gelling agent, this mixture is passed through a narrow orifice which allows the formation of the beads having the encapsulated explant material. Now, these coated explant material or encapsulated explant material are dropped into the complexing solutions such as calcium chloride. Now, we have coated the explant material with sodium alginate and we have dropped that encapsulated material into the calcium chloride solution. Now, this will allow the exchange of monovalent sodium ions by the divalent calcium ions. Now, once the monovalent sodium ions are replaced by the divalent calcium ions, the ionic cross-linking occurs. Now, once this ionic cross-linking occurs, this will harden up that encapsulated material. And this hardening of the encapsulated material or the formation of hardened coat around the encapsulated material or a polymeric box around the coated material is sometimes also called an egg box. Once the cross-linking occurs, the polysaccharide gets converted into polymer and this will further allow the encapsulated material to withstand different kind of stresses. And the third and the last step is the rinsing. Once the complexation is over, the hardened material is then washed with distilled water to remove excess of residues of chloride and sodium ions, which help in enhancing the shelf life of the material. So, this is the diagrammatic representation of the formation of synthetic seeds. We took the explant material, embedded it into the gelling agent. This gelling agent was passed through a narrow orifice to form the beads. These beads are then put into the calcium chloride solution. Last step is the rinsing and eventually we get the synthetic seeds. Now, synthetic seeds can be single coated and they can be double coated or layered. Many a times it has been observed that the single coated synthetic seeds, if they are stored for longer duration, then the coating material, that is polyethyl alginates, they form a permeable membrane around it which will cause leaking of nutrients and even dehydration of the somatic embryos. And therefore, double coating with a very thin layer of additional gelling agent helps in sustaining these synthetic seeds for the longer duration. But one has to optimize the thickness as well as the concentration of the gelling agent for the double coating. Now, we move on to the applications of synthetic seeds. It helps in preservation of germplasm of elite plant material like coffee or sandalwood. It also, these synthetic seeds also help in vitro propagation of endangered, rare and commercially important plants. They also help in multiplication of transgenic plants, because once the embryo has been modified genetically, that can be encapsulated and we get the synthetic seeds of that particular transgenic plant. They also help in germplasm preservation and they help in maintaining genetic uniformity and varieties of the crop. So friends, this was about synthetic seeds. Thank you.